Oh, I decided I'm going to go ahead and post the, I'll record it on Friday and post the extra lecture over the weekend. Because I didn't think anybody would be able to get it done by the time we got this morning. Yeah. So enzymes of glycolysis in eukaryotes are going to be organized into complexes. The substrate channeling, this is going to be the process that facilitates the movement of the substrates and products between enzymes. So this increases enzyme efficiency. It prevents the release of any toxic intermediates. Think about it like this. What would happen if I had a factory, it produced toxic stuff, and then I shipped it out every time I made that? And I had to ship it to a factory that's like 100 miles away. Eventually, I would spill that toxic stuff, right? And that's why toxic spills happen all the time, right? What's a better way of doing that? I put them all on one campus, right? Yeah. Hey, now I don't spill toxic stuff all over the place, right? Okay. And that's the same thing that's happening here. It also increased efficiency. What if I had to exit and re-enter a city every time I needed a widget, a screw? I would never get anything done, right? Because it takes so long to do it. And so these both greatly help these enzymes actually do their functions. And this is one of those reasons I kept going, this is not actually different stuff located in different places, happening at different times. Okay? These pathways are highly integrated and correlated together. Okay? Because they're oftentimes assembled into complexes in the same area and are actually portions of each other. Okay? The tumors display enhanced rates of glucose uptake and glycolysis. Aerobic glycolysis, this is the Warburg effect. This is the process by which the rapidly growing tumor cells metabolize glucose to lactate, even in the presence of oxygen. The 2,18F, 2D-deoxyglucose, abbreviated FDG. The abbreviation is so much easier to deal with. This is going to be a non-metabolizable glucose analog that's detectable by a combination of positron emission tomography. That's PET scans, by the way, in case you're ever wondering what PET scan was. PET scan's a stupid fucking way to say it, though, because tomography is a type of scan. Okay? And then CAT scans, which is the correct way of saying those, is also a stupid way to say it because computer-aided tomography is what a CAT scan is. Okay? Shit, made me say something stupid. Dumb and their stupid, correct way of saying it. Anyways, this right here is the pre-therapy versus the post-therapy. And you can see there's a considerable difference in these visualizations by FDG and positron emission tomography PET. That would actually be the correct way to say that, by the way, but it's not actually the correct way because MDs, I guess. Are you guys okay if I just say PET, or do I have to say the PET scan? Okay, you'll be okay? Okay. Anyways, you can see here, again, this is the pre-therapy, and we can see a lot of uptake here. See that okay? Yeah. And so over here, what are you noticing about that uptake? Greatly reduced or almost disappeared, right? And so post-therapy, you've hopefully killed that tumor, okay? Hopefully it's not just deactivated and it'll come back, but at least I hope for that person. The aerobic glycolysis, again, generate the lactic acid. The acidification of the tumor environment is actually going to facilitate the tumor invasion. The lactate is going to impair the activation of the CD8 plus T and the NK immune system cells that would normally attack that tumor. And so this is part of what's a problem with tumors. Tumors actually would not be as harmful as they are if they did not have this continuous production of lactate. This continuous production of lactate acidifies their environments, killing off other cells around them, and making, in general, all of their functions to produce energy, to do everything else, metabolism, etc., not work as well, right? This also ends up inhibiting the normal activities that would kill off the cells of the tumor, right? because we have an acidified environment. So the increased glucose uptake and the G6P formation provide substrates for the NADPH generating pentose phosphate pathway. Typically, this is, bless you again. Okay, this is typically denoted by things like 5-pp. Okay, why would that be 5-pp, by the way? What does pentose mean? 5? 5 sugar, right? P, phosphate pathway. Okay. So as solid tumors grow, the O2 in their environment is going to fall. The use of the aerobic glycolysis is going to reduce the dependence of the cell growth on the anaerobic glycolysis, is going to produce the cell dependence on the cell growth on O2. Okay? And this is why tumors basically automatically switch to that pathway, is because tumors will actually use up all the O2 in their environment. And this is one of the other things that causes so much problems with the rest of your cells around them. Okay? This is why, like, if tumors and cancerous growths did not do these things, you could grow a hell of a lot of cancer and not have any real problems. Okay? Has anybody ever seen the people that grow huge tumors and they look like they've gained like a 100-pound tumor? They literally, some of the tumors have been 100-pound. And the big thing that happens with that is they're either in places where they don't really affect the other cells that are important, and so it doesn't really matter, like your stomach fat, or they're in places where they're not harming anything that's important physiologically. Okay, they may be harming your arm, for example, but they're not harming your midsection where I have important stuff, or your brain. So the hypoxia-inducible transcription factor, this is HIF1. This is a transcription factor that facilitates the aerobic glycolysis. This is going to increase the expression of glycolytic enzymes, glucose transporters, and signal molecules. Cancer and anaerobic exercise training will activate the HIF1. Yes, you read that right. Technically, cancer would be just like you doing the anaerobic training, where they put like masks or they go up into the mountains and train. Yes. What a weird life we live in, right? Vascular epithermal, endothelial growth factor. This is VEGF. This is a signal molecule that's going to facilitate the growth of blood vessels that will provide nutrients to your cells. These are going to be the proteins in the glucose metabolism that are going to be encoded by genes regulated by hypoxia-inducible factors. And here we have the GLUT1, GLUT3, you may remember those from before, the hexokinase, the phosphofructokinase, that one we just did in the previous section. The aldolase, glyceraldehyde 3-phosphate dehydrogenase, the phosphoglycerate kinase, enolase, and pyruvate kinase. This right here is going to again be that overall pathway as denoted graphically to make it easier to understand. So this is the hypoxia again, altering that gene expression in your tumors. So the hypoxia, what does hypoxia mean again? Hyperoxia would be more of, right? Okay. The hypoxia is HIF1 is going to be then activated by that hypoxia. This is then going to go on. It's going to cause blood vessel growth so that we can have more supply coming in. But also it's going to cause the metabolic adaptation. This is going to increase your glycolytic enzymes activities and the amount that you have of them, of course. And this is going to allow your tumor to, one, have more blood vessel growth in the area so you can get more and more nutrients. And two, to activate so that when it's low on oxygen, it can still get energy and make the metabolic pathway derivatives it needs. Okay? I acted like that was a positive thing, but For the tumor, I guess it's positive. For the human or animal it's probably a negative. Okay? Though it is normally a positive if you need it, like if you did not do this stuff, when you get low oxygen environments sometimes in your arms or brain or whatever, it would not adapt to it. Okay? So it actually is a positive normally, just not in this case. Gluconeogenesis, if you will look at that term, what do you think that means? Gluco as in glucose, neo as in new, right? And genesis as in God created heaven and earth sort of stuff. So Genesis, creation. The synthesis of glucose from non-carbohydrate precursors. This converts the pyruvate into glucose. This occurs mainly in the liver with a small amount in the kidney and other tissues. This is why actually you are going to end up having most of your storage around these areas. And that's why you have a lot of the buildup in those areas. Okay? But also, if you ever go on like a ketogenic diet, by the way, you'll end up making your own sugars, and so you'll need to do gluconeogenesis. By the way, do you have to eat any sugar in your diet at all? No. No, the amount of carbohydrates in your diet you need, 0. Absolute 0. Don't need a single one. Okay? You can make all of the carbohydrates in your body, period. Lipids, not true. Proteins, not true. Okay, there's amino acids and lipids you cannot make. But carbohydrates, don't need any. That's why people can actually do things like the lion diet or ketogenic diets, and they can be long-term healthy and survive. And in fact, there's some really crazy research showing that those diets can actually deactivate certain types of autoimmune diseases and actually cure them. They don't get reactivated. So, weird. Who knows what the fuck's going on there? But anyways, apparently fasting also can do it. So it's probably that same pathway. Anyways, fascinating research, very new, like last couple of years. So this right here is why you can do those, but if you do them wrong, if you do not actually do them in a proper manner, very carefully, you can cause liver damage. Okay? That's why. Kidney and liver damage, because you'll build up toxins in those areas. Because those are the primary areas that make your sugars. Yeah. So this will convert pyruvate into glucose again. The important during starvation and fasting, because glucose is actually the primary fuel, and in some cases, in some parts of your body, the only fuel that will be accepted. And again, it's the only fuel for red blood cells, primary fuel for the brain. There's actually a few other places that are less whole organish, where it's the primary fuel as well, or only fuel. But these are whole systems. Did you know that? Red blood cells, only fuel? Yeah. That's why if you get sick, especially if you're feeling really down in energy, you can take some sugar and it'll actually help the process. Because your red blood cells can be regenerated more faster. You may be low on that. Though most people are not low, at least Americans. We are like never low on sugar. Okay, so anyways, the gluconeogenesis precursors, the major precursors for the gluconeogenesis are going to be lactate, amino acids, glycerol. These are going to be converted to pyruvate, or they can enter the pathway at later intermediates. Pyruvate is the traditional head of the river, the head of the pathway, okay? Because traditionally we anchor this as if it's the reverse of glycolysis, okay? Which makes a lot of sense because it's very similar to the reverse, okay? And then what we do is we have the other pathways in being the things like glycolysis where you can branch out, right? So it ends up looking like a mirror image. But again, that is human-centric idea, right? Understand that it could be entered via these different areas. So the lactate dehydrogenase is going to convert lactase into pyruvate. The triacyl glycerol hydrolysis in fat cells will yield glycerol and fatty acids. Animals cannot convert fatty acids into glucose directly. Glycerol is going to enter either the gluconeogenic or the glycolytic pathway as dihydroxyacetone phosphate. Remember the dihydroxyacetone phosphate? It's the one that interconverts back and forth. Yeah. Okay. That's where the glycerol enters. What part is the glycerol again? Typically the head, right? Then you have the fatty acid tails, which is the majority of the energy. Okay. Notice again, what did I say? The fatty acid tails, majority of the energy, do not go directly into. They can't go directly into glucose. Okay? They have to go a pretty extended pathway, actually, to be able to have anything to do with glucose at all. Okay? This, again, is the glycerol. This is the head. And so what you're going to do first is you're going to take ATP. This is going to be in the presence of glycerol kinase. That gave you a big hint. What's it going to do to glycerol? Yeah, phosphorylate it. You got it. And so we get glycerol phosphate. Note that this is symmetric, glycerol is, and so which one you're doing this on, it doesn't matter at all, okay? Just not the middle. The next step is going to be NAD plus going to NADH and NAH plus. This is going to be the glycerol phosphate dehydrogenase. You are doing what again? Dehydrogenating glycerol, yeah. Meaning stripping some H's off it. This is going to mean you make a carbonyl here in the middle. So one of these H's here comes off and one of those H's. And that makes the carbonyl bond. Again, that's an oxidation there, isn't it? Okay. And this gives you the dihydroxyacetone phosphate. Notice again, that's that DAP, right? Okay. This right here this is the pathway of the gluconeogenesis spelled out in all its glory. Notice, by the way, this is where the glycerol comes in right here. This is your amino acids, well, some of the amino acids at least. This is lactate, and this is some other of the amino acids. By the way, this is the pyruvate. This is the traditional head for the gluconeogenesis because that's the traditional output of the glycolysis, right? And we've lined it up to be the exact same way as the glycolysis, just in reverse. See that? Okay. Notice that these equilibrium steps for glycolysis, guess what? Yeah. Same stuff, right? The ones that have reverse steps that are not going to be the exact same thing are going to typically be these committed steps, right? Because they're committed. We won't be able to reverse that easily. Okay? Note, again, these are those red steps where we start having divergences happen. An interesting one being we actually use GTP here, which is woo, exciting. Okay. By the way, note we again have done ×2 here. Why do we do ×2 here? Yeah, to get a full glucose, we would have to go 2 times. Do we have to use lactate twice? No. We could do one lactate giving us one of the pyruvates. We could have an amino acid giving us another. We could have a glycerol giving us one of these, and the pyruvate gave us the other, the glyceraldehyde 3-phosphate. And they assemble to a glucose. They don't have to be, they're interchangeable. The 3 irreversible steps in glycolysis, these have to be bypassed by gluconeogenesis because that energy barrier would not normally be able to be easily overcome, right? Instead, essentially, we would just end up halting at those steps. And so we have to bypass and go around the mountain that is that energy barrier. And so this is the glucose plus ATP with the hexokinase. This gave us the glucose 6-phosphate plus ADP. And again, that was only going one direction because it was a -8 kilojoules per mole, right? Or kcal per mole, excuse me. -33 kilojoules per mole. The fructose 6-phosphate plus ATP. This is the phosphofructokinase, by the way, that we had just talked about a little while ago, which was one of the major regulatory steps. This gave us the fructose 1,6-biphosphate plus ADP. And again, that's that large energy barrier. We're not going back over the mountain the same way. Okay? And again, this one right here, this is the phosphoenol pyruvate plus ADP with the pyruvate kinase. This gave us pyruvate plus ATP. This one in this case is actually the phosphoenol pyruvate. It's just so high energy that it's difficult to regenerate it using ADP, right? It's hard to go up that table. Remember our table of the different potential energies for these different phosphate compounds? Yeah. It's very hard to go back up that. So we need some sort of energy coupling to get that over. So our pyruvate carboxylase is going to catalyze the carboxylation of pyruvate to oxaloacetate. This is going to use a molecule of ATP, and thus we have coupled it with ATP to push that up that energy barrier, right? And make something higher energy. So this is going to be pyruvate plus CO2 plus ATP plus water. Again, we have pyruvate carboxylase. That indicates what? You're going to take pyruvate, you're going to add a carboxyl group. Yep, you got it. And you can actually see that we added a carboxyl group. That's nice. By the way, this right here is getting added over here. Everybody see it okay? And this is ADP plus an orthophosphate plus 2 H pluses. The carboxobiotin, this is going to link to the lysine. This can act as a movable arm that carries your CO2 groups. The carboxylate group of the biotin is actually going to be linked to the epsilon amino group of the A-specific lysine residue by the amide bond. So right here is biotin, by the way, which actually I think has become popular in like health and beauty things, right? Anyway, biotin. This is going to be the carboxybiotin. Again, it's going to have that covalently bonded here to the amine. See that amine group right here? Bonded to the carbonyl. That was the carboxylate over here. Okay. And this is then going to be the epsilon amino group of your lysine. And so that's the lysine right there. That's the amino acid here, right? Everybody see that okay? Okay, so the carboxylation of your pyruvate is going to take place in 3 stages. Right here we have that CO3 minus plus ATP. This is going to give you your HOCO2 and the phosphate group on it, plus ADP. The biotin enzyme then, which is what we were just looking at right here, we bound the biotin to the enzyme, so now we're using it. This is then going to be added. Anybody know what that is, by the way? That's the acid form of CO2. Okay? That's what happens when you make it into an acid in water. Okay? And this is how the majority of your CO2 is actually transported in your blood. It's carbonic acid. Okay. Or I should say the conjugate base of that acid. More correctly. Okay, so we've bound that to a phosphate group that's higher energy now. This is going to help us push this reaction through. And so what we're going to get here is the CO2 now is going to go ahead and bind to that biotin and the enzyme. This is why that's that CO2 transportation arm. Okay? And so we put the CO2 on that. We're going to get back an orthophosphate group. This is where that extra oxygen went, by the way, in case you're wondering. Okay? The extra oxygen, hydrogen here, that's where those went. Okay, and so the CO2 bound onto the biotin enzyme arm is then going to react with pyruvate. This is going to give you your oxaloacetate. This is actually going to, the pyruvate carboxylase is actually going to function as a tetramer that's composed of 4 identical subunits, each of which will contain 4 domains. The biotin carboxylase domain, which is the BC, biotin carboxylase, Technically, it'd have a domain behind it. This is going to catalyze the formation of the carboxy phosphate. It'll attach of the CO2 to the biotin carboxyl carrier protein domain, which is your BCCP. Yeah, they do the right number of C's. The BCCP, this is going to be the one that swings to the active site of the carboxyl transferase domain. That's the CT, which is carboxyl transferase. The CT is going to be what transfers the CO2 to the pyruvate to form oxaloacetate. The pyruvate carboxylase tetramerization domain, which is just PT here, which the T is the tetramerization, P is pyruvate, facilitates the formation of the tetramer. This is going to be the binding site for your acetyl-CoA. It's going to be required for the allosteric activator. This right here is that whole thing that we just talked about with their different subunits right here. And so this right here is that biotin carboxylase right there that we just mentioned. This is the biotin carboxyl carrier protein that we had just talked about up here. And then this is the pyruvate carboxylase tetramerization domain. That's where you're going to form that tetramer. Okay? You can kind of remember this because it ends up being in the middle so that you can form that tetramer. Then we have here the carboxyl transferase. This will be the one that actually transfers it. The malate dehydrogenase is going to catalyze the reduction of the oxaloacetate to malate. Malate is going to be what transported from your mitochondria to the cytoplasm by a cytoplasmic NAD plus linked malate dehydrogenase. It's going to be reoxidized to the oxaloacetate. If you reoxidize this, by the way, that's going to be what yields your NADH instead of the NAD plus linked malate. And so you get that NADH back. The phosphoenol pyruvate deoxykinase, that's PEPCK, this is going to be what decarboxylates and phosphorylates the oxaloacetate to the phosphoenol pyruvate. The phosphoryl donor in this case is actually GTP, which is kind of a unique thing, and also helps you anchor this back into what we were just talking about up here, because that's right there, right? Those are the sort of things that will help you put these things together, by the way. If you ever have biochemical pathways you need to trace out without me, which you will someday. The oxaloacetate, this is used in the cytoplasm for the gluconeogenesis. It's going to be formed in your mitochondrial matrix. And so right here we actually have our mitochondria and our mitochondrial matrix right here inside. And again, this will be your cytoplasm and your cell here. And then that's the area between those. This right here is your pyruvate. This is going to have your ATP and CO2, going to ADP and orthophosphate. And that's going to give your oxaloacetate. That's happening in the matrix. The NADH plus is going to go to NAD plus, and that's going to give you your malate. Your malate is actually what's transported out. We haven't really touched on this yet because we haven't had a chance, but there's a shuttle, the malate shuttle, and that's doing that. Okay? Our malate then is going to be out in the cytoplasm. This is where we're going to have the rest of that gluconeogenesis happening, which is going to take NAD plus to NADH, NH plus, and give us our oxaloacetate out. And we're back in our gluconeogenesis pathway. Okay? Anybody have any questions about that so far? Okay, fantastic. Okay, so here's our oxaloacetate. This is going to be the one that uses up GTP. I'll help you find that if you ever need to go back in the overall pathway. The phosphoenol pyruvate carboxykinase is going to be transferring that phosphate and putting it on the oxaloacetate. This is going to be what gives you your phosphoenol pyruvate. Now, note again that this step right here is pushing against that potential energy table, isn't it? We're going from GTP, which is energetically basically ATP, right? And we're going up to a higher energy one, which is phosphoenol pyruvate right here, GDP and CO2. So the sum of your reactions that will be catalyzed by the pyruvate carboxylase and the phosphoenol pyruvate carboxykinase is this right here. This is your overall sum of reaction. And it's pyruvate plus ATP plus GTP plus H2O gives us phosphoenol pyruvate plus ADP plus GDP plus a free phosphate, the orthophosphate, plus 2 H pluses. These reactions are going to be used to bypass that pyruvate kinase reaction in your glycolysis because, again, this would not be energetically favorable to try to climb up that. We would be going upwards on that potential energy table. And the phosphoenol pyruvate is going to be metabolized by the enzymes of your glycolysis in the reverse direction until the next irreversible step. And that's why the coloring on these steps, by the way, in case you're wondering. Okay? So at this point, we've gotten to here, right? And from the next steps on, it's just a reverse glycolysis, isn't it? Okay? The next one, we have to, again, worry about this and diverge into a new step is this committed step again, right? This high energy monodirectional glycolysis, monodirectional gluconeogenesis step. Okay? Okay, so the allosteric enzyme fructose 1,6-biphosphatase is going to catalyze the hydrolysis of fructose 1,6-biphosphate. This is going to be 2-fructose 6-phosphate and the orthophosphate. And that's the overall reaction right there. So the phosphatase is going to be an enzyme that catalyzes that hydrolysis of the phosphate to the form of the inorganic orthophosphate. The difference between the kinases and phosphatases is going to be whether the phosphatate is transferred to water or ADP. That's the difference between them. If we are regenerating or taking it off of ATP, that's going to end up being the kinases, right? And then if we have the phosphatases, that's going to be where we get the waters. Okay? And we just throw water on there. The generation of the free glucose is going to occur primarily in the liver. The glucose 6-phosphate is going to be transported into the lumen of the ER. The glucose 6-phosphatase is going to hydrolyze, excuse me, hydrolyzes the glucose 6-phosphate to glucose. The enzyme is going to be bound to the ER membrane, in fact. The glucose and the orthophosphate, these are going to be shuttled back to the cytoplasm by the transporters. And so, yes, we're kind of shuttling these things all around. Part of that is to trap them so we can increase concentrations and drive reactions forward in these different steps. Okay. In most tissues, the glucose 6-phosphate is going to be converted into glycogen for storage. And so you'd have a little bit of local storage, and you'd build up your storage of glycogen. But if you need the glucose, you'd keep going along gluconeogenesis. Okay? At this form, though, this is actually easier to go ahead and convert to glycogen right here. Right? Otherwise, you'd have to do the glycogen, that reaction again to get to that same phosphorylate one. This right here is that uptake, and right here is the cytoplasm. Here's the ER lumen. Here we have the T1 transporter. And so we're going to have the glucose 6-phosphate. This is going to have a hydrolysis occur where we regenerate the free phosphate and we get the glucose. Okay. And then the T2 transporter is going to be what transports that phosphate back. The T3 transporter is going to transport the glucose. Notice we have a complex here with those transporters with the glucose 6-phosphate and the free phosphate being exchanged. Okay? What is it complex to? The glucose 6-phosphatase, which is what transfers the water here onto this phosphate to get the orthophosphate. And again, that phosphatase, what did that tell us? If this is a kinase, that would be ADP, wouldn't it? Yeah. It's not ADP, though, is it? Yeah. And the reason that's not ADP is we're chopping that phosphate off, and we're not re-getting that energy back, are we? Okay? We need this to be relatively energetically favorable in one direction. Okay? This direction. Anyways, this is then going to get transported back out into the cytoplasmic side here. Your formation of your glucose from your pyruvate, this is energetically unfavorable unless it's coupled with favorable reactions. If we were to try to do this process, the stoichiometry of gluconeogenesis being this right here, what we can see is that because of the additions of these ATPs, GTPs, and NADHs here, the delta G for this overall is negative 48 kilojoules per mole. If we were to look at the stoichiometry of the reversal of your glycolysis, so instead we just used the glycolysis pathway, we did not avoid those high energy steps, we just barreled through them. That actually is a plus 90 kilojoules per mole. And that's probably not going to go backwards very fast, is it? Or at least very often. Okay? And that's a huge energy difference in the wrong direction, isn't it? Which again, do you know how we can get those, by the way? So when we're talking about energy differences, those are state functions. So what we can do is we can just flip the sides, can't we? So that's that same energy difference for overall glycolysis, just reversed. Okay? Okay, so this right here is the totality of the reactions of gluconeogenesis and their delta Gs. And we had the same sort of thing for glycolysis, but we've changed out a few steps, right? And what we can see here is that we actually do have some very high energy in the reverse directions. For example, the phosphoglycerate plus ATP yielding the biphosphoglycerate right here. This is actually very high energy in the opposite direction. So you really have to push that one through. But we do have downstream and upstream some energy that's pushing those along towards that. Okay? So the gluconeogenesis and glycolysis is going to be regulated within a cell such that one pathway will be relatively inactive, whereas the other one is highly active. Wouldn't make much sense to continuously just convert back and forth in between, right? Okay, so what do you think those signals are going to look like? If a signal activates glycolysis, what would you expect from it? What will it do to gluconeogenesis? Yeah, hopefully it inactivates, right? If a signal activates gluconeogenesis, what would you expect from it from glycolysis? Yeah, hopefully we're inactivating it, right? Now, it's not a perfect analog because, again, those committed steps are different in many of these places. But overall, that's the trend, okay? So you can only self-check yourself when you're thinking these things through by using that. So again, glycolysis can predominate when the energy or glycolytic intermediates are needed, when you're low in concentration on those important glycolytic intermediates or energy. The gluconeogenesis predominates when there's a surplus of energy and when you have a lot of glucose precursors, i.e., I could assemble these into glucoses. Now, keep in mind, we had just talked about the glucose 6-phosphate and how that can be diverted off onto making glycogen. So, to some degree, this is true, but on some degree, we're also talking about this being the entry to glycogen storage, right? Because if we have a lot of precursors, but we also have a pretty decent amount of glucose, we're going to get shunted off into glycogen. Okay? Okay, so the interconversion of fructose 1,6 biphosphate and fructose 6-phosphate, this is going to be one of those key regulation sites. When energy is needed, AMP inhibits the fructose 1,6 biphosphate and is going to stimulate the phosphofructokinase. By the way, that's that same one we had just talked about stimulating before. Okay? Now we're just adding the fructose 1,6 biphosphatase, which would be the reverse reaction, right? Our same step, but gluconeogenesis one. When the levels of ATP and citrate are high, the ATP and citrate are going to inhibit the phosphofructokinase. The decrease in AMP is going to relieve the fructose 1,6 biphosphate inhibition. So this is going to turn off glycolysis. This is going to turn on the gluconeogenesis. Again, the reciprocal regulation at that interconversion of your phosphoenolpyruvate and pyruvate, this is going to occur in your liver primarily. The pyruvate kinase is getting inhibited by the allosteric effectors ATP and alanine. The pyruvate carboxylase and phosphoenolpyruvate carboxykinase, these are going to be inhibited by ADP. The pyruvate carboxylase is going to be activated by acetyl-CoA. This right here has a much better summary of it in a figure form. I find that far, far easier to understand and remember. Okay? Maybe that's weird, but I think that's pretty much everything. Okay, so right here, this is the glucose here going to pyruvate, of course, and here we have the glycolysis on the left and gluconeogenesis on the right, helpfully labeled as blue for glycolysis and this orange color for gluconeogenesis. Okay? Now note that these blue arrows right here, those are those glycolytic steps that didn't change. Okay? So we're just going to ignore those for this because they aren't important. The important parts being this right here is the glycolysis step. This would be the equivalent or reverse direction step for the gluconeogenesis. Remember, they are not the same pathways, though. They do not have the same, at these steps, the same enzymes. So this enzyme here, which is PFK, you may remember that the F26BP activated this. High AMP versus low ATP activated this because the high ATP would have inactivated, right? Reduced the activity. And so citrate also would reduce the activity of the PFK. Now over here, what we see is the gluconeogenesis. This is actually going to be inactivated by F26BP and AMP. So if we have a lot of AMP, we're actually going to inactivate this. Why would it inactivate from AMP? What does high AMP levels mean? I'm running out of energy. Yeah, so when you're running out of energy, do you want to use up all your energy? Making glucose? No, that wouldn't make much sense. Why isn't this activated by ATP? Yeah, this isn't an energy production thing, is it? So you actually are not as concerned about some parts of this, okay? Having a lot of AMP means you're low energy, right? And so don't use up all my energy making glucose. Or the other thing you could be doing here is you're not necessarily making glucose in this, are you? Because over here, in the steps that aren't really being shown, we can shunt over to glycogen, which is the storage of glucose. And so all we're really looking for here is, hey, am I low on the stuff to do glycolysis, or do I have a lot of stuff I could assemble and store? Okay? We're not really so much concerned with, hey, do I have a lot of ATP? Well, great, I have a lot of ATP. That doesn't mean anything. I could still need to store glucose, right? Okay, and so that's why these signals are not 100% analogous. You have to remember that these pathways are not 100% the reverse of each other for their purpose. Okay? energy production, glycolysis, gluconeogenesis, making glucose, but also it could be making glucose to be stored, right? Okay, so over here again, we have these bunch of steps. They had equilibriums. Congratulations, that's beautiful, that's wonderful. Over here on the glycolysis, we have the pyruvate kinase. This is going to be activated by F16BP. This is going to be deactivated by ATP and alanine. And again, this would tell us, hey, we have lots of energy, and hey, we have lots of alanine. We don't really need any more pyruvates made. Quit it. Okay? Over here, we actually have this broken up into several steps because this is a very high energy barrier to overcome, right? And so we needed to diverge and break this up into multiple steps so we didn't have to go over to the Himalayan mountains all in one go. Okay? And so this is the pyruvate carboxylase enzyme. This can be activated by the acetyl-CoA. Why would that be activated by acetyl-CoA? What's this next step after pyruvate? Does it just go into citric acid cycle? No, it generates acetyl-CoA, right? And so acetyl-CoA is the one that builds up if you are processing a lot of pyruvate, but before the citric acid cycle would use it up. This would be inhibited by ADP here. This goes to oxaloacetate. The enzyme that would then take that oxaloacetate to the phosphoenol pyruvate. This is right here, the phosphoenol pyruvate carboxykinase. This is actually going to be deactivated by ADP. Okay? And that's more of a case of I need the ADP for this reaction. Does anybody have any questions about those? Activation and inhibition? Activation and inactivation, I actually should say. So in mammals, the glycolysis and gluconeogenesis in your liver are actually going to be controlled heavily by the hormones. They're sensitive to the blood glucose concentrations. Your rates of glycolysis and gluconeogenesis will be adjusted in your liver to maintain your blood glucose levels. This is then for your red blood cells, your brain, your liver, and your heart all need this, though your liver can do it itself. The F26BP is going to stimulate the phosphofructokinase. This is going to inhibit the fructose 1,6-biphosphatase. When the blood glucose is low, your F26BP is going to lose a phosphoryl group. And so instead of F26, we're going to just get F6P because we lost one. And the F6P is not an allosteric effector of your PFK. And so it will stop messing with this because the F6P is not one of these, is it? See that? Yeah, we have avoided that, definitely. So the bifunctional enzyme, this is going to contain two enzymes on a single polypeptide chain. Bifunctional enzyme containing the phosphofructokinase 2, which is PFK2. Note, by the way, PFK, PFK2. Okay? Derivative. This is going to, and the fructose biphosphatase 2, this is the FBP-ACE2. This is going to determine the F26BP levels, which will then regulate this overall pathway right here. Because if we can lower or change, increase and decrease these levels here, we can increase and decrease the activation and inactivation of this. Okay. Okay. So our F26BP concentrations are going to be regulated by two enzymes. We have PFK2. This is going to form your F26BP. And then we have FBPase2 that's going to degrade your F26BP. So if we want to have an activation where we have an amplified activation, we would have PFK2. It's going to form the F26BP. F26BP is going to do what? It's going to inactivate gluconeogenesis and activate glycolysis, right? And so we're going to start taking more and more glycolysis, chopping it down, and getting pyruvate from that. Okay? And if we do the reverse, we start taking away F26BP, we're going to start activating glycolysis less, and we will start inactivating gluconeogenesis less. Okay? Meaning that our relative activities change towards gluconeogenesis. In case you're wondering what the summary of that was. Okay, so this is the bifunctional enzyme again, phosphofructokinase 2. We have two distinct domains on this. This is the kinase domain, this is the phosphatase domain. Remember that kinases and phosphatases are very similar, right? Okay. What does the kinase do again? Well, they both are going to do phosphorylation if you look one direction, right? But the difference being the ADP, okay? Here, we're ATP to ADP, right? Kinase. Here, orthophosphate. Water to orthophosphate. Okay? Did that make sense to everybody? Okay. And so this will contain an N-terminal regulatory domain. This is your N-terminal regulatory domain right here, by the way. It's kind of hard to see on this stuff, but it would be in the N-terminus. And a kinase domain, right there, yellow, and a phosphatase domain, red. And then that would have the carboxylate group over here, which would be the end. The PFK2 and SBACE2 activities are going to reciprocally controlled. This is by phosphorylation of a single serine residue. So when glucose is scarce, the glucagon rises. This is going to trigger a cyclic AMP signal cascade. This is going to lead to your phosphorylation of your bifunctional enzyme. This is by pKa, which is, remember, pKa is the one we commonly we're looking at, the 17 receptor domains. Everybody's okay with that? They're kind of remembering? Okay, good deal. This is going to activate your FBACE2, and it's going to inhibit your PFK2, or your PFK2. A kept coming in. Or maybe that was an needed a vowel. The pyruvate kinase, this is going to lower your F26BP levels. Gluconeogenesis is going to predominate in lower F26BP levels. When glucose is high, your insulin is going to rise. This is going to trigger a signal pathway. This is going to lead to dephosphorylation of your bifunctional enzyme. This is by the protein phosphatase. This is going to activate your PFK2. It's going to inhibit your FBPACE2. This is going to increase your F26BP levels. Glycolysis is going to predominate. Remember again that glycolysis does not necessarily mean that you're going to go all the way through, okay? You can actually end up shunting things off into other pathways because you're going to build up the equilibriums in all these steps down here. Okay. All right. This right here is a summary of that. So remember, these two are linked. So all of that was happening on the same thing, right? The same polypeptide. And this is one of the reasons why proteins, enzymes, and polypeptides are, yes, to some degree they are interchangeable, but to some degree they are separate concepts, right? So we can have a single chain that has two different enzymes, right? Depending on which one's active, because they have completely different sites. Okay. So this is the FBACE2, and again, this is the one that's going to be activated. We have glucose being scarce, glycolysis is inactivated, and this is how it actually does that inactivation right here. So we have the fructose 2,6-biphosphate being hydrolyzed to give us the orthophosphate and a fructose 6-phosphate, which does not do a PFK stimulation. The protein kinase A is the one that takes ATP, phosphorylates this, and that favors the FBPase2 activity. This one right here is the one that's the phosphoprotein phosphatase right here. This is actually further activated by the fructose 6-phosphate, by the way, which is what that meant. This is actually going to hydrolyze that phosphate, and that's that difference between phosphatase and kinase again, by the way. We're seeing that same sort of difference again. This is going to decrease the activity or inactivate the FBPase2 and activate the PFK2 portion of this by taking that phosphate off. And again, this is going to be where glucose is abundant. Your glycolysis is going to activate, so you can chop down the glucose into other ingredients. And so again, that's going to take that fructose 6-phosphate. We're going to put another phosphoryl group on there. And that's ATP going to ADP, again, that kinase, right? And again, that's going to stimulate the PFK, and that PFK is actually going to take the fructose 2,6 biphosphate, and that's going to move that through the pathway in glycolysis. Okay, anybody have any questions about that so far? No? So glucagon and insulin, these are going to be also used to alter gene expression by changing the rate of transcription. To stimulate gluconeogenesis, glucagon is actually going to inhibit the expression of 3 regulated glycolytic enzymes. This is going to stimulate the production of the phosphoenolpyruvate carboxykinase and fructose 1,6-biphosphatase. To stimulate glycolysis, insulin actually stimulates the expression of phosphofructokinase and pyruvate kinase, and the bifunctional enzyme that makes and degrades the the F26BP. The substrate cycle, this is going to be a set of reactions that will be in a loop. So the example of that is the phosphorylation of the F6P to the F16BP, and its hydrolysis back to the F6P. Keep in mind, even though it wasn't presented as a loop right here, that reaction is a loop in and of itself. Everybody see that? This is this down here, right? This one's this one over here, isn't it? And so that was its own cycle, its own loop. Okay. The reciprocal regulation is going to prevent both reactions from being active at the same time. Futile cycles, these are substrate cycles with a limited degree of cycling. These may result in pathological conditions like malignant hyperthermia. May be biologically important in enhancing metabolic signals. So small changes in the rate of the 2 opposing reactions actually will result in large changes in the net flux. Have you ever heard of habit is what you are? You ever heard that saying? Okay. What is the reason for that saying? Little changes build up to huge things, right? If you do 1,000,000 little changes, you end up with a mountain, don't you? Okay? And so this is the same thing. If we have a small imbalance in these two, eventually this is going to end up as a huge difference, right? It's only a matter of time. And so this is actually what we're seeing right here. Here we have 2 different parts of the same cycle, and we have a 10 imbalance, right? So for every 90 times we go backwards, we go forwards 100 times. So the net difference is only 10. This right here would be a net flux of 48 in this case, 120 versus 72, right? That changes how fast you would get a net increase or a net reduction, right? Alongside the rate of these reactions. You can think of this a bit as an imbalanced equilibrium. Does that make sense? If you look at this entire thing as an overall equilibrium, then right here we have an imbalanced equilibrium that's heading towards this direction, right? And that imbalance, that rate difference, is that 10. Okay? Okay, the Cori cycle, this is going to be a series of reactions that's carried out by the cooperation between the liver and the muscle. So lactate is going to be produced by the active muscles, and it's going to be released into the blood. It'll be then converted to glucose by the liver. Liver does the vast majority of that conversion. So you may have been wondering, Dr. Clausen, what the hell? We have lactate, it's in the muscle. That's a different place than the liver. Does Dr. Clausen think the liver and the muscle are the same place? And the answer to that is, I'm not no biologist. No, it's not that. It's that your blood connects the two, right? So we have to have some form of transportation and shuttling across cell membranes. So the nitrogens from amino acids used by the muscle for fuel are actually going to be transferred to the pyruvate. This will form alanine. You may have wondered why alanine had anything to do with any of these cycles up here. Did anybody ever wonder why the hell alanine was involved in this? No? Okay. Well then, here we go. So again, nitrogens from your amino acids will be used by your muscles for fuel. They're going to be transferred to the pyruvate to form alanine. The reverse reaction is then going to take place in your liver and build those back up into the amino acids that you just used. And so this is the Cori cycle as summed right here. So what ends up happening is we have glucose and lactate, and those are the ones that will go through the blood. Okay? And you'll find those in the blood, and they'll be transferring back and forth between your liver and your muscles. So the liver is going to release glucose. The glucose will go into muscles. The muscles will do activity. They're going to use up that glucose, convert it to pyruvate. If they don't get a chance to use that up, it can go into lactate, right? Which is that quote-unquote our version of fermentation, right? Okay. Or another way of saying that, that's that anaerobic process, okay? And again, that's going to produce approximately 2 phosphates worth over here in the muscles. Again, this is that glycolysis pathway going into the lactate fermentation pathway that we talked about. So our lactate's going to build up and around our muscles, right? Is that necessarily a good thing? Do our muscles just continuously keep working no matter how acidic and how much lactate gets built up? No, like the ethanol fermentation, we would actually kill ourselves. The muscles would die eventually, right? And so what's going to happen is the muscles are going to release that lactate into the blood. The blood's going to end up flowing throughout our body. It will end up going to the liver. That liver is then going to pick up that lactate, okay, and shuttle it into the liver. That lactate is then going to go through the gluconeogenesis pathway. Now it's going to do that by going lactate to pyruvate, and then the pyruvate is going to end up picking up 6 phosphates, and those are going to go to glucose eventually. Again, that's that gluconeogenesis pathway. Now keep in mind that the Cori cycle does not actually necessarily have to complete a cycle, okay? So if you ended up exercising a bunch, produced a bunch of lactate, your body's still taking that lactate and converting it back to glucose, you could end up having it and then you eat a bunch of sugar. You actually could end up going over here into the glucagon, okay? Which is one of the reasons why you don't necessarily want to go a little bit of exercise, then eat a candy bar, right? With a bunch of sugar, okay? Anyways, we got the complete pathway for the Cori cycle. Anybody have any questions about that so far? Okay, so during a sprint, you're going to have a cooperation between your glycolysis and gluconeogenesis that will occur within your muscle tissues, but you're also going to, in the long run, have a cooperation that will occur through the Cori cycle and your liver. And so right here, we have the muscle cell. It's taking that glucose. We're going to the glucose 6-phosphate. This is going to go to pyruvate. If we have the oxygen, we're going to go to CO2 and water, right? And that's going to provide us a tremendous amount of energy. But if we don't have the oxygen, we're going to have to go the anaerobic pathway. That means the lactate, okay? Now, keep in mind that you do have some glycogen in your muscles. There is a bit of there. And so you will have interconversion that happens between your glucose 6-phosphate and your glycogen in your muscles. So if you have excess glucose 6-phosphate, it will go to glycogen. And if you have a shortage of that because you're burning through your energy, you will, if you have a sustained enough run, start burning that. Okay? And then that's going to have the lactate. This is going to start building up in your muscles. They'll release it to your blood. Your blood is then going to distribute it all throughout your body. Now, what does that mean? Does that mean that goes directly to the liver? Congratulations, you get to pass prison and go? No. Your blood goes all throughout your body, right? And so it is going to go to your cardiac muscle as well. Okay? Now, that lactate, that can get converted somewhat in your cardiac muscle, but also can be converted to pyruvate in your liver cell. Now remember, your cardiac muscles, if any muscle is going to have oxygen in your body, which one's going to have it? Yeah, your cardiac muscle. It's right after you take in the oxygen, right? And so if anything's going to have oxygen in your body for muscles, it's your cardiac muscle. Okay? So what does happen quite a bit is the lactate does end up going into your cardiac muscle, and this can either be converted into glucose along the gluconeogenesis if your heart's not working that hard. But if your heart's working pretty hard, it can actually convert it into the pyruvate and then go along the normal citric acid cycle, and then we can get the CO2 and H2O. Okay? So it can basically use the scraps from your muscle cells. Does that make sense? Okay. Now, if you're probably going to end up having extra lactate that your cardiac muscle cells are not going to be able to use, that will splash over into your liver, or your cardiac muscle cells could just not be able to take up that amount in that amount of time, or it can even start running out of oxygen. So this is then going to plug into your liver, where we had talked about the Cori cycle completing. Okay, and so the pyruvate plugs in your liver. Now, your liver does actually need energy, and so some of that pyruvate does tend to get used up to make energy, okay? And that's just the normal citric acid cycle right here. The other thing that can happen is pyruvate goes to a variety of different precursors for different things like amino acids, etc., and so that can go to that as well, okay? Now, if you're running low on your glucose, you can actually start digesting other things. For example, you can take amino acids and you can start converting them into the pyruvate or the glucose 6-phosphate right here, okay? And those can get plugged in to make glucose. You can also take your glycogen stores and start using those up. Now, these are the majority of your glycogen stores in most people is around their liver, okay? That's the beer belly, and that's why that's the hardest thing to get rid of if you ever try to exercise and, get a six-pack or something, okay? So this gives you the glucose 6-phosphate, bless you, which will go to glucose, and then that again gets plugged back into your blood, and the Cori cycle keeps going, right? Anybody have any questions about that? Okay. Now keep in mind your glucose in your blood does end up, some of that goes into your liver, okay? And eventually you may actually have to regenerate some of this like glycogen stuff. So I feel like Americans, these arrows are probably really wrong for like your typical American that's drinking way the fuck too much sugar, right? And exercising way too little. Yeah. Which I'm guilty of too, don't get me wrong. It's not an outside statement, it's an inside one. The inborn errors in metabolism. So genetically inherited deficiencies in your activity or regulation of specific enzymes. Some examples of this are the triose phosphate isomerase deficiency and the pyruvate carboxylase deficiency. So TPID, this is going to be a multi-system disorder. It's going to result from dihydroxyacetone phosphate accumulation in cells, especially red blood cells to be very specific. So the presence in early childhood and may lead to death. The symptoms are going to include congenital hemolytic anemia and progressive neuromuscular disorder. This right here is the specific pathway that they're having issues with, the specific part. And again, remember that's that triose phosphate isomerase. That's that one that went back and forth between DAP and the G3P, okay, or the GAP, right? The GAP. Everybody remembers that one? It's at the very middle of the glycolytic pathway, and gluconeogenesis for that matter. Okay. So this is a deficiency right here. So when your triose phosphate isomerase activity is missing, half of your carbons for your glucose, these cannot be metabolized to yield the ATP. So you miss out on half the energy. Research actually suggests that the disruption of your energy metabolism is not the cause of TPID symptoms. If that was true, if this was the only problem, then you would just be you'd have to eat more, right? Get more glucose, no problem, right? But it's not. Yeah, that's not the major cause of your symptoms for TPID. So it's actually the buildup of the dihydroxyacetone phosphate that's going to cause your TPID symptoms. It cannot be converted into the methylglyoxamol. So this being the methylglyoxal. Methylglyoxal is actually a highly reactive molecule, has double carbonyl here, which gives a big hint that this is very reactive, that will covalently bind to available amino acids on proteins. This will yield advanced glycylation end products, or AGE. The modification is going to inhibit protein function, and obviously then you're going to have difficulty making any proteins, including the proteins you would need to grow big and strong and, survive your childhood. Yes. By the way, would eating more glucose help you with this condition? No, it'd just make things worse, wouldn't it? Yeah, because you make both of these. If you can never convert this back over to what the gap, you just keep building that up and making more and more of this, wouldn't it? Is there something that could help with this condition other than giving the person the enzyme? What if you ate zero sugar? Yeah, not easy to do as an American, but people do it. There's the carnivore diets and stuff. So if you ate zero sugar and were very careful on your diet, you could actually reduce the effect of this disease quite a bit. Yeah. Because the gluconeogenesis does not have to actually make the dihydroxyacetone every single time. But you'd have to eat a really careful diet. It would be almost impossible to do. Okay? So I would not, that's not obviously not the, yeah, that'd be incredibly difficult because there's amino acids that, if you go back up to the table of all the things that can convert it into that, right? It's not low. And you can still go back and forth in that pathway, right? So it would have an effect, but not as much of an effect as you probably would want. Okay. So PCD, this is going to be a rare disorder that's characterized by some extent by hypoglycemia and lactic acidosis. May lead to death in the first months of life in severe cases. Symptoms are going to include lethargy and seizures. So this right here is the pathway, pyruvate carboxylase deficiency, PCD. So obviously you're deficient in this right here, which is your enzyme. And you may recognize this because this is the pyruvate plus CO2 going to the oxaloacetate. That was that sort of first step of gluconeogenesis, wasn't it? And so you're going to essentially not be able to do gluconeogenesis properly. Pyruvate carboxylase is going to be, again, that key regulatory enzyme in gluconeogenesis. It occurs primarily in your liver. When the pyruvate carboxylase activity is missing, the liver cannot maintain adequate blood glucose concentrations. It cannot remove lactic acid from your blood, which would normally be used as a gluconeogenic precursor and plugged into that Cori cycle through that pathway. Okay? So you could exchange and recycle that to form new glucose. Your glycolytic enzymes with similar properties do not have similar amino acid sequences. Most likely, they're derived independently rather than through gene duplication. Glycolysis is going to consist of metabolism of the hexoses and metabolism of your trioses. The metabolism of your trioses is going to be common to glycolysis and gluconeogenesis. The enzymes in the metabolism of trioses are going to be present in all species. Enzymes in the metabolism of hexoses are less conserved. And this is why you get a lot of things where your 6-membered sugars typically are the ones that get people sick because they're less conserved enzymes. 3-membered sugars, on the other hand, everybody keeps those enzymes. Everybody has them. All organisms have those. Okay? All right. Anybody have any questions on that so far? Well, that's the end of the chapter. So anybody have any questions on that chapter? Nope.